What if I told you your brain doesn't just, rest when you're asleep?
Hmm,
What if it's actually busy, like really busy? Running a kind of sophisticated nightly car wash, meticulously scrubbing itself clean?
that's a great analogy, actually.
For years, we figured, okay, sleep is vital, memory repair, all that. But it turns out it's also this crucial, very active detox process.
Hmm.
So today, we're taking a deep dive into some really fascinating insights. They come from a recent research spotlight in trends in immunology. It's called Rest and Rinse. Sleeping rhythms drive brain detox.
Aptly named.
Right. And our mission here is basically to unpack these incredible, kind of intricate mechanisms behind how sleep cleanses the brain. We'll highlight some surprising facts and think about what it all means for our health.
And this brain cleansing, it really is extraordinary,
Yep.
Unlike most other tissues in your body, the brain's main functional tissue, the parenchyma, it actually lacks lymphatic vessels for fluid drainage.
No built-in plumbing like the rest of us.
Sort of, yeah. So the brain has essentially co-opted cerebrospinal fluid, CSF, for this job. It's a unique system, this CSF-based cleansing. It's known as glymphatic or glial lymphatic clearance.
Okay, glymphatic. And we've known for a while the system kicks into high gear during sleep.
Exactly. Most active during sleep.
But the specifics, the how of sleep triggering this cleanup, that's been a bit fuzzy.
A bit of a mystery, yeah.
But now, and this is where it gets really exciting, a recent study by Hogland and their team has kind of pulled back the curtain. They've shed significant new light on exactly how this whole process works.
It's quite a breakthrough.
So let's jump right into that core discovery from the Hogland study, how the brain's own rhythms, neuronal and vascular, actually cooperate to cleanse it.
Right.
It's not just theory anymore. They've shown this sort of stunningly precise choreography.
Yeah, and the really groundbreaking aspect here was the approach Hogland's team used. Very innovative, working with mice.
What did they do differently?
Well, they developed these cutting-edge, non-invasive methods. It let them measure several things simultaneously. Chemical signals like norepinephrine, blood flow changes, the movement of CSF tracers, even brain activity with EEG, and these tiny head-mounted microscopes.
Wow, all at once.
All at once. And crucially, they did this without head fixation. That's huge, because head fixing really interferes with natural sleep in these kinds of studies. This was natural sleep.
Okay, so a much clearer picture then.
A much clearer picture. Their work built on earlier findings about this small but really important brainstem structure called the locus coeruleus.
The norepinephrine factory.
Exactly. It produces norepinephrine, or NE, and they showed that during sleep, the locus coeruleus generates these ultra-slow rhythmic waves of NE, about 0.02 hertz, really slow, pulsing through the brain.
Ultra slow waves of any. Okay, so the brain's own chemical signals are setting the rhythm for this cleaning cycle.
That's the idea.
And here's where it gets, well, really remarkable for me. The NE, you said, is a potent vasoconstrictor. It narrows blood vessels.
That's right. Potent vasoconstrictor.
And Hogland's team found these NE surges actually came before the rhythmic shrinking and swelling of blood vessels, the vasomotion during NREM sleep, the non-rapid eye movement stage.
Precisely. The NE waves seem to drive the vasomotion.
So the chemical signal makes the blood vessels pulse?
Exactly. And if you connect this to the bigger picture, you can think of it like a bellows mechanism.
A bellows, okay.
Yeah, these rhythmic changes in the blood vessel diameter, the vasomotion, they're tightly linked to the movement of the CSF in the spaces around them. As the vessels constrict and dilate, they act like bellows, literally squeezing and pushing CSF along those tiny channels, the peривascular and subarachnoid spaces.
So the pulsing vessels physically move the fluid?
It seems so. The study clearly showed that the changes in CSF movement directly correlated with this vasomotion.
Okay, let me try and summarize this for you listening. Essentially, during sleep, activity in the locus coeruleus creates these slow NE waves.
Mm-hmm.
This NE then causes large rhythmic changes in blood vessel size, that's the vasomotion, and this rhythmic squeezing and stretching, it supercharges the exchange of CSF with waste products out in the brain tissue, effectively rinsing it all out.
That's a great summary. It's an active pulsating cleanse, like you said, a car wash for your neurons.
Amazing. And this cleansing isn't just about if you sleep, but how you sleep, right? You mentioned sleep architecture.
Yes, absolutely. The specific stages and cycles of our sleep, the architecture, seems to be vital for proper waste clearance. It's not just about the total hours you're in bed.
Which brings us to something pretty surprising they found about a common sleeping pill.
Right. This raised a fascinating and maybe slightly concerning point when they looked at zolpidem.
The sleeping pill, what happened?
Well, even though zolpidem did allow the mice to sleep, biologically speaking, it severely disrupted those crucial NE oscillations.
No.
And as a result, it significantly reduced the movement of those CSF tracers into the brain parenchyma, into the main tissue.
So less cleaning, even though they were asleep.
Exactly. Less cleaning. Interestingly, this happened even though zolpidem actually increased the frequency of microarousals. These are brief awakenings often driven by NE surges.
Huh. So the NE surges were happening, but not the right kind of rhythmic wave.
It seems that way. The research indicated these brief microarousals weren't actually required for the CSF clearance process itself, but their presence alongside reduced clearance might be a kind of warning sign, maybe a bellwether for glymphatic dysfunction.
That's really intriguing. Does this mean that just feeling rested after taking something like that isn't the whole story? That some sleep aids might actually be hindering this vital cleaning process?
It absolutely suggests that possibility. I
Yeah.
mean, the critical takeaway here is that it strongly indicates proper sleep architecture. A right sequence and type of sleep stages, not just duration, is what's truly crucial for restorative sleep and efficient waste clearance.
So just being knocked out isn't enough.
It appears not. The right kind of sleep seems paramount for your brain's nightly rinse cycle.
Okay, stepping back a bit. This has broader implications too, right? Beyond just detox. You mentioned connections to our immunological health.
Absolutely. This work also sheds light on other vital functions of sleep.
How so?
Well, it's likely that different sleep stages actually perform distinct, maybe complementary, functions related to waste clearance. Their specific sequence, their duration, that might be incredibly important.
Like a multi-stage wash cycle?
Kind of, yeah. For example, NREM sleep with those perivascular space oscillations we talked about. That could be the stage for really thoroughly mixing the CSF with the waste products in the brain tissue.
Okay, the soaking stage maybe?
Maybe. Then REM sleep is associated with profound vasodilation. The blood vessels widen significantly. This could compress the perivascular space, effectively pushing that now waste-laden CSF out of the main brain area, maybe towards the dura mater, that outer protective membrane, for eventual lymphatic removal.
So NREM mixes, REM pushes out. That's fascinating.
It's a plausible model based on these findings. And another key takeaway is how intricately sleep links to our immune functions.
Right, the immunology connection.
Yeah, CSF isn't just carrying waste. It also contains brain antigens and cytokines, which are molecules that can regulate immune responses.
Okay.
So by driving these brain antigens out during sleep, maybe it's giving immune cells patrolling outside the brain a vital chance to monitor what's happening inside the central nervous system, kind of like immune surveillance.
Makes sense. The guards get a report during the night shift.
Something like that. And we do know the mammalian immune system is generally more active during sleep than during wakefulness. Plus, remember those catecholamines like namilocane-nephrine?
Yeah, the ME waves.
They're also potent immunomodulators themselves. They can directly influence immune cell activity. So it's all interconnected, the cleaning, the immune surveillance, the chemical signaling.
And this research, it feels like it has massive relevance for long-term health, especially when you think about, well, neurodegenerative disorders.
Absolutely. Conditions like Alzheimer's and Parkinson's diseases, they are very closely associated with the accumulation of waste products, like protein aggregates, in the brain.
And also with sleep problems, right? That connection's been known for a while.
Exactly. Persistent sleep disturbances are a common feature. So understanding this fundamental mechanism of waste clearance during sleep is, well, potentially huge.
It really clarifies why sleep is so critical in that context.
Precisely. This study, it really paves the way for entirely new ways to study how sleep architecture is involved in clearing waste from the central nervous system. Those NE oscillations, the vasomotion, they give us new mechanistic handles, things we can potentially measure and maybe even influence.
So new targets for research and maybe therapies?
That's the hope. Developing strategies that could perhaps improve restorative sleep, maybe enhance this cleaning process. Ultimately, this kind of fundamental research might uncover entirely new therapeutic avenues for treating these devastating diseases.
That's incredible. But I have to ask, the study was in mice. How confident are researchers that this exact mechanism, the NE waves, the bellows, translates perfectly to humans? And what's next?
That's an excellent and important question. Well, the fundamental biological principles, fluid dynamics, cellular functions are likely quite similar across mammals. The precise nuances, the exact frequencies, the scale might differ in humans.
So more work needed there.
Definitely. Further research will absolutely focus on validating these findings in human studies, perhaps using advanced, non-invasive imaging techniques like specific types of MRI to try and observe vasomotion and CSF flow during natural human sleep. This mouse study gives us a fantastic blueprint, a really detailed model, but now we need to build on it and confirm it in people.
Understood. So let's try to recap our deep dive for everyone listening. Sleep. It's not just passive rest, not by a long shot.
Not at all.
It's a crucial active process for brain waste clearance. And it seems to be intricately driven by these slow norepinephrine oscillations coming from the locus coeruleus, which in turn drive rhythmic vascular pulsations, vasomotion.
That's the core mechanism they uncovered, yes.
And the key takeaway isn't just how long you sleep, but the quality of that sleep. Specifically, the sleep architecture, the right cycles and stages seems paramount for this nightly detox.
Absolutely. Quality over just quantity, it seems, for this particular function. Understanding these fundamental mechanisms truly opens up new frontiers, not just for potentially treating neurodegenerative diseases, but maybe more broadly for optimizing overall brain health for everyone.
There's still so much to learn, isn't there, about what our brains are up to while we're dreaming or just sleeping deeply?
Definitely. And this research gives us powerful new tools and concepts to keep exploring that.
Okay, so here's a final thought for you to consider. Knowing how intricately our brains seem to be cleansing themselves during sleep, this whole rest and rinse cycle, what small shift might you consider in your own sleep habits to potentially optimize your brain's nightly rinse cycle? Something to think about.